you want to do the other one? We have all of the needles laid out so that when he's fully under anesthesia, we can start fish surgery, where we'll put 2 of the needles at the halfway point in red and white muscle, and then 2 of the needles towards the caudal end in the red and the white muscle. Once the needles are in, we will attach these little clips to them that will allow us to plug it into the computer, and that way we can transduce the signal better. How's he doing? Good. And now that all of the electrodes and stuff are set, we're just going to put the fish in the tank. We'll get a baseline reading where we'll just run the software and make sure it's picking up on all channels fine, and then we'll start with our experiment. Here we go. Okay. So we have our GoPro camera set up right here. We're just making sure it's all set to the right settings. And then once both Megan and I are good, we'll start running the experiment. Hey buddy, you want to swim? Please? For the nice people watching at home. We're doing like 12 trials a minute a trial. 12 minutes? 15 minutes?
Yeah, I was thinking 15.
Yeah. But like if he's all good after this experiment, like physiologically and all of the wires and stuff are still transducing signals, we'll just leave him in and change the temperatures.
Yeah.
We'll try to get another experiment in.
Yeah. Oh, I see a little.
Yeah, there's a little shift in there. There's a little flutter at the end. So bigger movements will create bigger peaks, which we will be able to see better, but seeing as Jigglypuff is being a little shy right now, it's probably still under the anesthesia a little bit. We're just getting very minimal signals. So that LabScarf software is really cool. Basically what we do is we take all the channels that have significant data, so like if we don't identify it as noise or a channel came out, we will rectify the channels where we'll just find like the absolute value of all of those things, and then we will in the software tell it to find us the time 1, time 2, T2 minus T1, so like the duration of a burst, and then the max minus min, which will show us the intensity, and then we will manually go in, identify individual bursts, put the markers, and then store that data, and then from that we can get cycle duration, we can get duty factor, figure out like peak intensities, how many bursts they have like during movement, and from that hopefully we'll see that different temperatures affect them differently, whether that's in terms of intensity or more bursts, like more muscle recruitment for performance activity, all that kind of stuff. Go!